talk. And I haven't really prepared anything specifically, but I actually, first I should do is thank my graduate student, Colton Parker, because a lot of this presentation is something he made to present to the neuroscience program open house, where's our new first or prospective graduate students are coming in, and he prepared some of this. And a lot of the work that I'm going to show is stuff that he, data that he collected along with Zach Johnson, who I got to tell you a little story. There's probably no one out there that knows Zach. Gene is not here, but Zach was actually an undergrad here, like a long time ago in like 2007 or so. And then he went on to get a graduate degree with Larry Young, who many of you may know at Emory University. And now he's with Todd Streelman, doing some really amazing work in the cichlid fish, but also using this tool that I'll talk about today, this single nuclei RNA sequencing. And it's kind of technological. And I know that this is for the public as well. So I'm going to try to explain this tool that he's kind of introduced me to, and I'm totally blown away by it. I think it's an absolutely incredible tool that we now have access to. And so the main part of this talk is just to sort of demonstrate how awesome this tool is and how it can be used to really uncover some interesting biology that we didn't know before. And I hope that those of you who have other problems or things going on, scientific questions, that you'll sort of think about how you might use this tool, because it's really remarkable. Completely blown away by it. So thank you, Colton and Zach, for all your work. And basically, I'm just presenting what they've done here. So let's start out by talking about sex. So everyone likes talking about sex. So for me, though, I take a philosophical view about sex, standing back and thinking from an evolutionary perspective, what the heck is sex for? And when I think about it, I think, okay, well, it seems like it's important for generating variation, because that's fundamentally what it seems like it's doing, right? It's recombining genetic information from two individuals, potentially the same individual, but ultimately, it's two different individual genes, chromosomes mixing together and so on, making unique individuals. But if you think about it, though, that in itself doesn't really require there to be two sexes, because you can imagine making gametes that just fuse together, right? Why do you need to? And in fact, in microbes, this variation isn't caused by horizontal transfer. There are no sexes and so forth. So where does the sex come from? Is the solution that many organisms have evolved for producing this variation, mixing the genomes, producing the ideas in the changing environment you want to make to survive and persist the longest. You better have ways of making new recombinations and new phenotypes, and that's one way to do it. But the solution that's come up is that one sex or one component of the system makes the eggs and the other one makes the sperm. And therein lies an asymmetry, because obviously the egg for one is much bigger. And when you're making eggs, that involves different processes, both in the gonads obviously are different, but also the brain, like we'll talk about today, that's controlling your gonads and making them make eggs and sperm or sperm. That is an asymmetry that has to now flow through the entire organism and affects their behavior, of course, because if you're making big eggs, then you might have different priorities in terms of the resources you're devoting them, and so that you might be more picky about who you're mating with. And if you're a male and you have a lot of sperm, maybe you're less picky, and it affects whether you're defending a territory and how you might. So it goes on and on and on. This asymmetry sort of cascades down. And there are sexual dimorphisms we see all over the animal kingdom, right? And some of them are very dramatic, like I'm showing you among many birds. And this has to do with, of course, sexual selection for crazy features that females find attractive, that probably have no value other than they find them attractive, and it causes these differences. But we see sexual dimorphisms all over. In fact, humans probably are one of the least sexually dimorphic. We love looking at ourselves and we see all these huge differences. But if you imagine an alien looking down at the world, they probably, and I always think to a fish, certainly they can't tell the difference. And you look at a lot of fish like these species here in the low, where the males are brightly colored and so on, females are very different. So sexual dimorphisms are all over, and even in the animals that don't have obvious sexual dimorphisms like humans as much, there still are dimorphisms that must occur in the brain and must occur in the gonads for making eggs versus sperm. And so where in the brain do we see the biggest sex differences? And there's a whole lot of interest in this topic because males and females are different, as we know, and they do have features that overwhelmingly, obviously they're the same but there are certain parts of the brain that are different. Some of those parts of the brain are involved in regulating behaviors, but some of those parts of the brain, like I mentioned, have much more basic function, and that is that they regulate the gonads. And when you're making eggs and releasing eggs, that requires different regulation than when you're making sperm. And an area of the brain known as the preoptic area of the hypothalamus, for those people who have not studied the brain, probably haven't heard of that, but this is an area of the brain that is very important, not for cognitive processes like making decisions and thinking about, your emotions and things like that, but more doing autonomic things that you don't have to think about, like ovulating. You don't think about that, that just happens, or making sperm, or regulating your appetite, or growing, or when you're stressed and you have released stress hormones. These things are very reactive, but there's parts of the brain that still are controlling that. And in the hypothalamus, there's this particular brain area known as the preoptic area that has neurons in that part of the brain that directly control the gonads through regulating a gland that's right below the brain called the pituitary gland. The pituitary gland, of course, releases hormones that communicate to the gonads. People have heard of luteinizing hormone and follicle stimulating hormone. Those are hormones that are involved in regulating the menstrual cycle, but also they're found in males and females. And they are how the brain communicates to the gonads through the pituitary. And not surprisingly, this part of the brain that has neurons that control the gonads is sexually dimorphic. Males have different composition of this part of the brain than females because they have a different task that they need to do, which is regulating testes versus sperm. And this, by the way, this preoptic area, the neurons in these areas, the way they're regulated, the way the pituitary releases luteinizing hormone and follicle stimulating hormone, and the gonads and their composition are highly consistent across all vertebrate species. So all vertebrate species have a preoptic area. They all have neurons there that we can identify that regulate the pituitary gland release of the gonadotroph, the gonadotropins, the LH and FSH. So I'm interested in how these sexual differences develop. And, in one of the things that's really hampered this field is that a majority of what we know about how these sexual dimorphisms develop, number one, has been done in mammals. Why? Because we care more about mammals than other animals. But in mammals, the way it works is that a female development occurs, and I put in quotations by default in the sense that in the absence of any gonads, a female general body plan unfolds, right? I don't know if people know of a disease called androgen insensitivity syndrome. I was looking it up to try to show some people the slides of this, but this is where you are a male, you have Y chromosome. This is in a human syndrome, and you therefore develop testes during embryonic development, and you start releasing androgens, which is the male sex hormone like testosterone. But the thing is, the way hormones work, of course, is if there's no receptors for that hormone, it doesn't matter that you're making it. And people who have androgen insensitivity syndrome don't have functional androgen receptors. So even though they have testes and they're releasing androgens, none of their tissues know that. And so it's almost like they have no androgens, or they're not releasing androgens, even though they have a Y chromosome. And the phenotype of these human individuals looks female. They have testes that haven't been descended, and they have what more looks like female genitalia, a female body type. So the general plan, the idea is, and I don't know if I wanted to talk about this, and this is sort of philosophical thinking, but it makes sense that would be the general plan in the first place, because of course, female really is all that you really need to have in order to have a sustainable population. Or in the case of I don't know if people know, but there are many species of vertebrates which are parthenogenic, which basically don't have any males in the population. So females are capable of producing eggs, which are large enough to differentiate into a full-blown organism. So the idea is that it makes sense that female development might not require any gonadal hormones because there are many species that don't even have males in them. There are no species that I know of that are only males. Of course not, because you can't. So anyway, the idea is that the general plan is that female sort of occurs by default. It unfolds without any tampering by the gonads. And how do you get a male then is when you have a testis that's formed and that starts releasing androgens. Of course, you need the androgen receptors as well. But if you have that going, that changes the trajectory away from the female and you develop the male into the male. And because of this fact, a great much more is known about the process of masculinizing the preoptic area as opposed to feminizing, which sort of unfolds in absence of any signal. So the idea is we need to know more about the feminization process. Like even though it is default in the sense that it's not necessary to have gonadal hormones, it obviously there's still active and coordinated processes involved in, for example, producing 10 times the number of neurons in a part of the hypothalamus in females as compared to males. So that is a very active process that requires mechanisms. They're just not initiated by sex hormones. They occur and unfold by default. Now, so that's where we come in and we say, okay, we don't know a lot about feminization. It occurs by default. It's difficult to study in mammals because of that, because there's no clear signal like an androgen, which you can tie to and you can manipulate and study. Nevertheless, it occurs. So can we find a species where feminization occurs in departure from the male phenotype and in response to a clear signal? And is there such a species that exists? And as I suggested, the general plan might suggest that they would be rare because of the female being the general default, an idea of that. But here's where my species of fish fits in. My favorite fish, the anemone fish. Amphiprion ocellaris is a specific species I study. There are about 30 different species of these fish. Many of you probably know these fish live on sea anemones, which are animals themselves that are cnidarians. They're like upside down sea anemones. They have stinging cells on them, which, by the way, the stinging cells themselves are super cool. They're like little cells that have trap doors that if you touch them, they open a little trap door and a spear comes out and sticks into you and injects a little poison. And that's why they're sticky to the touch. But for some reason, the anemone fish don't get stung by them. And by the way, it's nothing like genetic or specific that they have. I think a lot of it is a behavioral acclimation, in a sense, to the anemone, but they're able to tolerate the stinging cells of the anemone, and then they seek protection. It's a great way of protecting the fish from predation. And in fact, people have looked at this and there's very low predation rates for anemone fish. And as a result of that, you might predict that based on life history theory, that an animal that has very low extrinsic mortality might live a long time. And that's indeed the case. These animals can live up to 30 years. And many people keep these in captivity. So we have very good estimates of that. I myself have some of these in my lab that are probably at least 25 years old. So they live a long time. They're also monogamous, socially monogamous and genetically monogamous. Of all the species, we all know that people talk about their monogamy and they're, at best, they're sort of semi-monogamous. And usually animals have extra pair copulations, but really not so for the anemone fish. They're very faithful. Once they develop these pair bonds, the only two that reproduce, and it's because they live in these very small groups. They live in, especially the species I study, the anemones they live in are far apart from each other. And there are only usually small numbers of fish on these anemones, like maybe 4 or 5 fish. And they're always in this way where the biggest fish is always female, the second largest is male, and then if there are other ones, they're smaller, and they're gamma, meaning they're subordinate. They don't have any role in the reproduction. And by the way, these fish are not related to each other. It's not like they're born on the anemone, like Nemo suggested, right? In Nemo, they had the little egg there. That's not what happens. Once they hatch, they go into the plankton cloud, and they there's 1 in a gazillion chance they make it because everything in the world is eating them. But if they make it, they settle out of the reef. And if they're lucky enough to land in a place where there's anemone and it's empty or enough to tolerate them. So in other words, these are unrelated, small groups, strict dominance hierarchy, strong genetic monogamy among the monogamous pair. But note that if one happens to die, like the female dies, it could take a while for another female to get there and so on, because they're pretty isolated in these groups. And as a result of that, there's a very unique feature that's developed where if the dominant fish croaks, then what happens is the male will change sex and become female. So that's pretty amazing, right? I mean, so this is an animal that is reproducing as a male. It's behaving as a male. There's lots of differences in behaviors between the sexes too. For example, the males do all the carrying of the eggs, or vast majority of the carrying of the eggs, fanning them. So that part of Nemo is correct. But the male will do most of the fanning and taking care of the eggs, plucking off debris from the eggs. And the female is usually the one that defends the territory against big intruders like humans. Like I've been times when I'm scuba diving and I see one of these big maroon anemone fish, and I'm a monster with my buddy coming up there to this territory, and the fish will come up and bite me. And they're very aggressive. I would argue they're some of the most aggressive. I've seen sharks underwater. They don't do that, right? Sharks will look at you, maybe go through. These fish come out and bite you. So thank God they're only, this big or whatever. So female is super aggressive, and she's the one that comes out and bites you. The male's there taking care of the eggs. So that has to change. Obviously, their gonads have to change. Obviously, their brain has to change from regulating male gonads to now regulating female gonads. I mean, this is an amazing, dramatic transformation. Again, the same genome, right? Thinking about it from that standpoint, the same genome is capable of producing totally different sexes, right? So now I want to shift to telling you about this new strategy I have, a new technique I have that we have available to us, and some of the discoveries we've made about the differences in the brains of the males and females in these fish. And that sort of sets the stage for what type of reorganization is necessary here. So, no one has really done this before. So we're looking now, and what I'm going to show you now are results from the single nucleus RNA sequencing that we did of 6 male anemone fish and 6 female anemone fish. And here we're looking at the region of the brain I was describing earlier, this preoptic area, this area of the brain where we know there are cells that regulate the gonads through innervating pituitary. And we also know that this part of the brain is sexually dimorphic in every vertebrate species that it's been looked at. And so it's likely going to be dimorphic in our species, but which cell types and how is really the goal of this study. So, anyway, how do you do this? This tool is unbelievable. So you take the brain out, dissect the brain area of interest. You basically dissolve, you have to mix up the tissue so that the cells are sort of separated. And then you filter it through a filter, keeping just this nuclei and all the little other stuff flow through. And you're keeping the nuclei separated from the rest of the junk that's in that cell, that tissue section or whatever. And then you can use some fancy tools to even purify those nuclei even more through sorting them and so forth. We've decided that you may not need to do that, but in this particular data set I'm showing you, we did do that, where you're basically further isolating just the nuclei. So at the end of the day, you have a little jar that's filled with nuclei and a highly purified sample of nuclei from your brain area of interest. Now, these are the nuclei from the cells that are in the brain. So they include neurons and glial, others, all different types of cells in the brain. There's also blood in the brain. So some of this will have blood cells in there as well. The blood is in blood vessels, right? That's penetrating the brain. And this will also be able to measure those cells. So then what it does is, this is the miracle of this technology, is each nucleus then, there's a machine that is able to take each nucleus, and I basically sequence a lot of the RNA transcripts that are in that nucleus, okay? And so then you have this very high dimensional data that has each nucleus, you may have measured 10 to maybe 1,000 different transcripts. These are RNA fragments, right, in that nucleus. And then what you can do is you can then, because you have thousands of genes measured in tens of thousands of nuclei, what I'm showing you on the right is a plot showing each dot is a nucleus, and its position and color on this graph represents its particular gene expression profile that it's displaying, right? Because certain cells will express, for example, and I'll show you in a minute, this population of cells right here, hopefully you can follow my cursor, happens to be a type of cell in the brain known as radial glial cells. And I'll show you that in a minute. But all these cells are related to each other and are expressing genes that indicate that they're radial glial cells. For example, there are certain things that radial glial cells and only radial glial cells express. For example, aromatase is a gene. So these cells are identified all together on this graph because they share a similar, we call it a transcriptome, which is all those different transcripts that it has in the nucleus. All these nuclei are together because they're all very similar, because they're all from cells that are astrocytes. And by the way, the axes on this graph are not important. It's called UMAP1 versus UMAP2, but UMAP1 is basically, those of you who are statisticians, I don't know how many people out there, but you can think of this as like a principal component or a linear combination. It's basically a reflection of a lot of different genes, but if you're far on one of this side, it identifies you as being separate and having a very different transcriptome from a cell that's on this side. So the further you are away on this graph, the more different those cells are. But you can see that it identifies very many different types of cells in this analysis. So here's an example of what we can do more with this analysis is you can identify then the classes, the different types of neurons and cells. So for example, based on the genes that these different cells express, I can identify, like this population over here, I know are glial cells and astrocytes. And I can know that these cells right here are microglia. I don't know if people have heard of those cells in the brain, but microglial cells play a very special role in communicating the immune system from the periphery to inside the brain. And you have macrophages. Remember I mentioned there's blood in the brain, and we can measure those cells as well. Here they are. They're oligodendrocytes. These are cells in the brain that form the myelin sheath. And so they wrap axons, and so we can see them. We can also differentiate the glutamatergic, the excitatory neurons that release glutamate, from the inhibitory neurons here that release GABA. And then there were some neurons that had both, which is amazing to me. It may not be the exact same cell, but some of the overlap may occur. So all this is to say is that this allows us a really detailed window into the different composition of this preoptic area. And next, what I'm going to show you, well, here's another way you can distinguish different cell types as well. For this audience, maybe it's not appropriate, but all what I'm doing here is just showing you that here's a population of cells that expresses galanin, oxytocin, arginine, vasopressin. I mention those because these are very well-known peptides that are involved in the regulations of social behaviors that tend to be different between the sexes. Like oxytocin is critically involved in caring for offspring. And as I mentioned, males are much more devoted to caring than females. So one might expect this population to maybe be sexually dimorphic between males and females. Just as a hint, it didn't turn out that was not one of the populations that was different, which is interesting. But anyway, there are tachykinins and various different neuropeptides that are shown as marking these different cell populations of interest. But here's something, look at this now. I want everyone to understand what this is, this graph at least, before we go. So, and at the top, what I'm saying is that this is evidence of major reorganization of the brain, of this part of the brain, that must occur during sex change because males and females have very different numbers of neurons of specific types. Let me zoom into one of these, which is this one right here. You see this cluster of cells? It's an inhibitory population of cells that is very numerous in males. So you can see there's a lot of cells there in the males. But in the females, that's basically absent. So here's an entire population of cells that we can now characterize, and we have, and I'll show you what these cells are. It turns out they're super interesting. This cell, very prominent in males, disappears in females. So during the process of sex change, there must be some mechanism by which this population disintegrates, either undergoes apoptosis, meaning programmed cell death, or maybe these cells somehow change their transcription so much that they end up some other place on this map. But in one way or the other, that indicates a very dramatic change. And this is in the direction, of course, where females are losing cells. And that's shown here in this graph by all the blue dots. And the darker the blue, the bigger the difference is in terms of more cells in males and females. So you can see there's that big one that I pointed to. Everywhere else, there's sort of a light effect that now we can talk later. People are interested. We think may it just be an inevitable consequence of the fact that there are a lot of populations of cells that are much more numerous in females than males. And that's what's showing you in red. You can see some in the middle here, where right in the smack dab in the middle, like these poles, these red poles here, where in the females are many, many more of those types of cells than there are in males. You can see this other population of cells here below, where there are many more cells in females and males. Likewise, up here, and these poles, this is glutamate cells, these are GABAergic down here, where there are more cells in the females than the males. So we actually looked at each of these clusters independently and tried to statistically compare and determine, which clusters look like they're significantly different in the proportion of cells that are found in females and males. And we came up with half of the clusters that we were able to identify. In other words, half of these discrete populations of cells that we were able to distinguish based on that transcriptome profile are sexually dimorphic in numbers of cells. That's extraordinary to us. So we were thinking maybe one or two populations. But that suggests, of course, we are talking about the preoptic area, which is a crucial area involved in regulation of the gonads. So it's not maybe too surprising, but the magnitude of the reorganization still is astonishing. And now I just zooming in on that one population here. This population that I mentioned that's sort of absent in females, that's present in males. That population of cells is super interesting. For one, it expresses tyrosine hydroxylase. And tyrosine hydroxylase is an enzyme which synthesizes dopamine. So that suggests that this population of cells that's much more numerous in males and females likely synthesizes dopamine. And it also expresses androgen receptors. That's what this androgen receptor is saying. So this population of cells seems to express dopamine and also androgen receptors. And it's interesting too, if you look at these two clusters, that it looks like they're almost different cells that express tyrosine hydroxylase and androgen receptors within this cluster, as if there's maybe two subpopulations within this cluster. One that's androgen receptor positive and the other one which is tyrosine hydroxylase positive, because it doesn't look like there's too much overlap there in these two markers. And I put up these markers here, DLX2A, which is another gene. The only reason why this is of interest is because this in other species has been shown to label cells that directly innervate the pituitary gonadotropes. It's the pituitary cells that release the gonadotropins, LH and FSH. So that then, combined with tachykinin 1, together suggests it's very likely that this population of cells that we've identified, that's much more numerous in males and females, likely is directly innervating the pituitary gland. Now in fish, those of you who know anything what I'm talking about the gonadotropins and regulation in mammals, know that in mammals, there is no direct connection between the neurons of the hypothalamus and the pituitary gland. There's a portal system, which means that those neurons release their hormones into a little local blood system that supplies those hormones to the pituitary. So it's not a direct innervation, but in fish are different. They have direct innervation of neurons in this part of the brain right onto the pituitary. And so it's very likely, we think, that this population of cells, this inhibitory population of cells that contains dopamine, is innervating the pituitary and thereby regulating the gonads, possibly inhibiting the pituitary from releasing a pattern of gonadotropins that causes a female gonad and phenotype, and having these cells present, continuously inhibiting those gonadotropes, keeps the system in a male phenotype and away from the female pattern. And maybe when you remove these cells, that unleashes a female pattern of gonadotropin regulation in the female gonad. Well, that's our one hypothesis. But anyway, I'm going to continue on here. Hopefully, I'm not losing anybody. I have no idea because I can't really see a lot of people, and I'm talking about a lot of complicated things. So if people have questions or anything, please interrupt me. Otherwise, I'll just keep going here. But another really cool thing you can do this technology here, and I've showed you this already, but this is a graph again showing you each dot is a cell, and its different place on this map is dependent on its transcription. So they're basically segregating these cells based on their function and their cell type. And what you can see here is just the different cell types that are in red are much more numerous. They're more numerous in females than males, and in blue, darker the color, they're more numerous in male than female. And so this is one way of thinking about the cytoarchitectural differences in the POA between males and females. Notice that these say nothing about the location within that brain structure. These cells could be distributed evenly throughout the brain, but they're just much more numerous in a female brain than a male brain. And another way to look at this dimorphism, in addition to the numbers of cells, is to look at which of these cell populations shows a different transcriptome, is basically functionally different in terms of its RNA readout, right? Certain cells that are functioning differently should have a different transcriptome, should have different expression of different genes if it's functioning differently in male versus female. So this is one type of dimorphism, could be that these cells function exactly the same way in males and females. In other words, transcriptome is exactly the same. There's just a lot more of them, right? And that's how it produces the difference in the regulation between the sexes. An alternative is that there's no difference in number of these cells, but that the cells in females just function very differently. They have a different transcriptome, possibly because they have receptors for steroid hormones and they're responsive to estrogen, possibly, maybe not. And so the next slide I'm going to show you is a summary of which of these clusters shows dramatically different transcriptome. And the darker the green means the more genes that we found were differentially expressed between males and females. So the darker the green means those clusters have a heck of a lot of genes that are different in males and females, are expressed at different levels, meaning that they likely are functioning differently, they're regulated differently in females and males. And I want to flip back and forth a little bit between the previous graph, which shows you again the cytoarchitectural dimorphism, to the transcriptome dimorphism, if you may. And you can see here that there's some of these, like particularly this one down here and this one up here, which are very dimorphic and more numerous in females and males, and also are extremely dimorphic in their transcriptome, more than between 200 and 250. I think this, I'm not sure if this, Colton can remind me if that means 200 to 250 significantly differentially expressed genes by a false discovery rate of 5% or something, right?
Yeah,
right?
that's right.
Got it. That's right. So anyway, first of all, you can see that there are a lot of them there. I want to just point out, though, again, because I know that there's at least a few people in the audience that I saw that are interested in some of the neuropeptides that are known to regulate social behaviors like oxytocin and arginine vasopressin. If you remember, I'm going to flip back here. Those are in this population right here. And they're, so if you think about this little island here, when you look at the graph coming up, you can see there's sort of the bottom pole there. There's a little bump right there. So if you go back to these differences here, first of all, you see that there's no, this is the bump right there. There are no differences in cell numbers between males and females for those neuropeptides. And there are no differences in transcriptome between those populations of cells, which was very surprising to me, given that these fish behave differently. So I still don't really know what that means. And I'm welcome to have people's input on that. But I should also say that that area of the brain was the area of the brain that had all the steroid hormone receptors. So we can also look at which of these populations have steroid hormone receptors. And it's that population, the oxytocin, galanine, arginine vasopressin cells, that showed absolutely no difference in transcriptome or numbers of cells, but of course have all the androgen receptors and estrogen receptors, which then begs the question, how in the heck could they not have a different transcriptome, since these are nuclear receptors and they have all of them, and males have a different steroid hormone profile than females? So I do think that is a little bit of a mystery here. And I haven't solved that, but it's just interesting that that's the case, and that a lot of these differences where you see are numbers of cells, totally different transcriptomes, do not have any estrogen receptors or androgen receptors. Now, I should say that the estrogen receptor ESR2 beta is actually expressed quite widely. And so that one could be mediating some of the effects. But in terms of, which populations have, AR1 and, have all the different androgen receptors, well, that's this one population that I mentioned before, which is super interesting to us because it's present in males, absent in females, and it happens to be clearly or very likely innervating the pituitary, regulating the gonadotropes, and has androgen receptors, which to me screams that it's involved in the maintenance of a male reproductive axis, where you remove these cells, then it unleashes a female reproductive axis. And I should say, now to really confuse the heck out of you, I'm going to show you these crazy models. And maybe what I summarize here is that what we have going on is, I was mentioning, is the hypothalamus, which has these cells that are regulating the pituitary gland. And the pituitary gland is releasing LH and FSH, and that's communicating to either the testes or the ovaries, depending on what sex you are. And as I mentioned at the beginning, of course, if you're a male and you have ovaries, you have to have different regulation of them. So you have to have a different pattern of release of the LH and FSH or something to maintain ovaries separately from testes. And that requires changes at the top, at the hypothalamic level. And what I've shown you is that in these anemone fish, there are dramatic reorganization that we see, where these dopamine neurons that have androgen receptors and tachykinin peptide that likely inhibit GnRH neurons and inhibit the gonadotropes directly are being removed, thereby unleashing potentially a gonadotroph female profile. But at the same time, you also have massive increased numbers of GABAergic cells and glutamatergic cells. Those were those populations I was showing you with the poles. And some of these may be inhibiting these gonadotrophs. Some of these may be exciting. So GABAergic cells are excitatory on GnRH cells. GnRH, of course, stands for gonadotropin releasing hormone, which is one of the populations of cells that regulates these gonadotropes, which, by the way, I didn't talk about this at all, because it's another one of those cells which is completely having us scratch our head, because it doesn't show any differences in sexual dimorphism, both in number of cells or transcriptome. So that's a mystery. But a lot of these other cells do show obviously dramatic differences and are likely regulating the gonadotropes. So this is the model we have, and we're super interested in the key role that these inhibitory cells may have in initiating this process of gonadal transformation in the fish. And one thing that we're very interested in exploring is, to what extent do all those brain reorganization that I just described to you, to what extent does that have to happen before the gonads even change, right? Because obviously, in order to get ovaries, you need to have the right type of signal. And so some of these changes have to occur before the ovaries develop. And but which one of these occur before the ovaries develop, and which one of these are dependent on the gonadal hormones, is really a key question that we have yet to answer, and is definitely top on our list of future directions. So future directions, and this is literally, I just copied and pasted the specific aims that are currently in my NSF grant that I'm about to write, that I'm writing. So hopefully these make sense at this point. But the first one is to determine if the neurosexual differentiation that I clearly showed you today in very dramatic terms precedes gonadal differentiation, okay? And we have some preliminary data that suggests that at least some of those differences, especially the populations of cells that multiply in the females, that those happen before the gonads change at all. And that makes sense because, of course, you need the change at the top to affect the stuff at the bottom. So anyway, then the next aim would be to determine the role of neurogenesis and apoptosis in mediating neurodifferentiation. I didn't have a lot of time to talk about this, but in mammals, the process of masculinization of the POA is known in exquisite detail in terms of the processes that are activated from the gonadal hormones stimulating the microglia, which then activates them, and they gobble up neurons so that you start off with a lot of neurons because you're on the female default, and then you gobble away those neurons, and that's how you end up with fewer neurons in the male AVPV, or that area of the brain I was talking about. In other places, there's neurogenesis, but we know nothing about these things in the feminization process. So to what extent is neurogenesis required for multiplying all those neurons that I was talking about? Seems pretty likely, I gotta say, I agree. But it is possible that some of those changes are occurring due to sync cells changing their transcriptome rather than de novo genesis and proliferation. So that needs to be explored, and the mechanisms for apoptosis and what's regulating this. And of course, all this stuff starts purely with social context. So it's the brain ultimately, signaling happening in the brain, that has to trickle down into affecting neurogenesis and apoptosis, that then has to trickle down into affecting the gonads. So the last aim is to really identify the function of that sexually dimorphic inhibitory population of tyrosine hydroxylase positive, androgen receptor positive POA neurons that I told you about, because we think they may be involved in this switch from testicular to ovarian pituitary regulation. And our idea is, and lucky for us, and this is another, again, I want to end like this because this is another like unbelievable thing you can get from a single nuclei RNA sequencing, because we've measured all these genes. We can ask, is there any gene that specifically labels this population and no other population? Because if we can find such a gene, we can use genetic strategies to drive like inhibitory and excitatory dreads, which half of you probably have no idea what I'm talking about, but half of you may, to actually stimulate or inhibit these neurons. But lo and behold, Colton found FRMD7, which no one in the world knows what that is, but it's a gene that apparently is expressed only in this population of cells. It's expressed a little bit other places, of course, but much higher and most visibly in this cluster, which then gives us a means to specifically genetically manipulate that population so that we can either cause it to fire action potentials or inhibit it from firing an action potentials. And one would hypothesize that if I inhibit it, that would be like removing it, which would be like unleashing the female gonadal trajectory, and we might be able to change a sex by manipulating the function of particular neuron populations in this part of the brain, which would be pretty cool and pretty and tell us a lot about obviously the circuitry and the differentiation differences. So on that note, I will end and take any questions from everybody, but thank you, Colton. I love you. And Sarah and Abby and Veronica and Emma, Leah and Prithika and Streelman Lab in Georgia. As I mentioned, I didn't mention this actually, but Zach is actually now currently, I did mention this. He worked with Todd Streelman at Georgia Tech. And he really helped us initially learn the tools, the technique, and he's been doing some awesome work with it and pushing it to the boundaries. And you should see some papers coming up here pretty soon where you're going to see some other fantastic things that you can do with this single nuclei RNA sequencing, including looking at activation of different cells, correlation patterns between clusters to look at connectivity, and it goes on and on. So we're really excited with this data set, but we're worried that we probably won't have to do any other experiments again and just continue to analyze this for the rest of our lives, because it is that deep. But I will stop there and take any questions if anybody has any.
Awesome, thank you very much. That was really interesting. I loved hearing about that. If anyone else wants to unmute and if they have any questions, or you can throw them in the chat if you want to do that. Either way, but with us. And I had a couple questions selfishly I can throw in there while we're waiting for other people. But I guess a couple things I had. So you talked about a lot of like transcriptional changes between male and female. Do you see morphological changes? Because you lose populations of those neurons. Do the brains actually look differently? Are there morphological differences, or is it just in transcription?
Yeah, great question. So actually, the first thing we did was do a very like a more classical histological analysis of the brain, and sectioned the brain of males and females, and stained the brain with what's called a Nissl stain, which just basically allows you to visualize the cell bodies. And then we counted numbers of cells in different parts of the preoptic area and measured the volume of different regions of the brain. And yes, we find that females have a larger region of the preoptic area. And this is common across all vertebrates, basically. And most of these cells usually are involved in regulating the gonadotrophs. We don't know that. All we know is that there are more cells in females than males. They have larger volumes in this particular region. So you can look at the brain and say, yeah, there it is. I mean, it's not like, you can tell a male and a female just from looking at the section. It's quantitative though, but they do have larger volumes and more cells. We also know that fish that are changing sex, that they actually get more cells in that area of the brain. And that process seems to occur at a very regular rate and is independent of their gonads. So their gonads basically don't change right away. They revert to like this static, intermediate state, and they just stay there. And they're just releasing a little bit higher amounts of estradiol than a male and a little lower amounts of this 11-keto-testosterone. So they're kind of intermediate, they just stay there. But meanwhile, the brain is changing. So that's why we believe that some of these brain changes are likely occurring independent of the gonadal hormones. Because in these fish, there's a lot of stuff happening in the brain in terms of volumes of brain areas changing when their gonads aren't doing anything. Their gonads are just basically shriveling up. So, yes, these are morphological differences. The other thing is that, I'm going to flip your question around, and I was talking to Colton about this, but, in the past, and even your way your question is formed, it's like we're used to thinking about anatomical differences as stuff that you can see. Look, there's a bigger nucleus. Oh, look, it's smaller over there. But what if the cells are actually 10 times numerous? They're just spread all over the brain. You would never be able to know that with that anatomical approach. With a single nuclei approach, you would be able to know that. So we don't know that the population that's 10 times as numerous, they may not be in one particular location. I do know that in the females that there must be a population that's clustered enough in one area and multiplied enough, because I can see that morphologically. But some of those differences may not actually reflect in a nucleus that's larger or smaller. They may actually be distributed evenly across the whole brain. Of course, the only way to know that is to do some kind of in situ sectioning of the brain and staining them for these different markers, which is certainly on the list of stuff to do. But yes, there are, the idea is that some of these are relate directly to like larger volumes, and some of them are even more interestingly maybe distributed, where we didn't even think of them before. We didn't even know to look for them. And now we have a tool that can unleash them and uncover them that we didn't know before.
That's really cool. The single nuclei, being able to look at differences in single cells is always super intriguing to me.
It changed the game a lot because, of course, we had transcriptomics, and I was always, I know this is an institute for genomic biology, so I don't want to say too bad things, but like, a lot of the genomic stuff led you more questions. Just you have long lists of genes and you don't know what to make of them because they could be in any mix and match of cell types. And the change in gene expression that you might see in response to treatment could be driven by having more cells of one type or the other, or more. You don't even know that. So here, it gives you a lot more information when you have the transcriptome within the cell, like exponentially more information, it seems.
And then another question that we had was just more of a general one, like, are sex hormone levels in fish, are they like steady state, or are they cyclical like in mammals? Do they levels change?
Yeah, they're cyclical. So we did a study that one of my grad students' first study we published on the anemone fish was just looking at sex differences in the steroid hormones. And male fish, the bioactive androgen 11 keto testosterone is obviously like orders of magnitude higher in males and females. It doesn't seem to vary very much, but males don't really have anything going on that makes sense that it would vary across the breeding cycle. Females have much higher 11-ketotestosterone levels. I'm sorry, estradiol, very low 11-ketotestosterone. And those estradiol levels vary across the breeding cycle. So they peak, at times when they're ovulating, or I don't know exactly what the pattern is because we haven't linked it up with what's going on with the eggs. But they definitely go up and down over the breeding cycle, which is not surprising. But
can I hop in?
hop in.
Yeah, thanks. This is super cool. Colton, I feel like I'm gonna hit you up for coffee and ask you lots of really obnoxiously detailed questions.
He's the guru for the single nuclei.
Yeah, well, I'm really curious about it. And so one thing, so did you say this was POA specific or you did single nuclei but whole brain?
We did. I wouldn't say it's POA specific, and I was going to show those slides, but I didn't do it because I thought I wanted to make it a little bit more informal. But Colton can share that with you. What we do is he basically takes the whole brain, puts it in a little dish that has like this RNA RNA ACE, so that the RNAs are stable, and dissects out part of the brain, like pulls off some of the lobes in some of it like a standardized way as possible, and shaves off some of the pieces. So he definitely gets all the POA and some surrounding areas of the brain.
Sure.
So that's what we'd say. And he's refined it a little bit over the time. Yeah.
Yeah, no worries. I was just curious. And so my other question is like, you're not seeing some of the like classic sex differences in like AVT, oxytocin. And so like, what do you think is going on?
I don't know. We do see differences in expression of the IT receptor, and it's higher in males and females. And that's what we found with real-time PCR in the whole brain. And we did find like higher aromatase expression, which is higher. That's not the IT cells, but beyond the actual expression of those genes, I would have thought that like the oxytocin cells would have a different transcriptome because because they're expressing the steroid hormone receptors, right? And so I don't have that. I really don't know. I want to talk to you about that. I want to talk to them. I don't really know why that doesn't make any sense to me. They have steroid hormone receptors, and I just told you a second ago that there are huge differences in the circulating steroids. So presumably those are binding to those receptors, and those are nuclear receptors, so they should be changing transcription. So those cells should have a different transcriptome, but they don't. Like not even close, like not even, right? It's not like it's didn't meet significance. It's like they're just like like they weren't different.
Yeah,
right?
I'll say this is getting into the weeds a little bit with the data, but at least with the isotocin cells, they're kind of concentrated in that one cluster, but they're fairly wide. So there's a lot of cells that are sort of outside that cluster as well too. So some of the kind of cluster resolution analyses that we're doing we need to do more layers of analysis that are just pulling those cells out and really targeting them. So there might be stuff there that we just haven't dug up yet. But like Justin said, it's like there's so much potentially to be done.
Well, actually, that being said, have we done a transcriptome difference between IT positive cells between the sexes?
I did that with aromatase. I did it with IT receptor expressing. I don't think I have done that with ICT cells, or if I did, it was a while ago, and I haven't looked at it in a while.
They're definitely not among the big one. They're not among the big hitters, though. So there are a lot of other cells there that are far more different in males and females that don't even seem to have any receptors. Like they have ESR2 beta.
Cool. It's super cool stuff, guys, and obviously a super cool system. So
yeah, thanks. Anything else?
Awesome.
Hello, hi Justin. Hey. That was a really cool talk.
Good to meet you. I've never seen
wanting to see you in person for a while.
I haven't seen you. I got to come and go and see you guys.
Yeah. This is all super cool, and I think a lot about sex change and what that means. I have a lot of questions, but I won't bug you with all of them. I am curious if you have any insight into there seems to be a ton of variation in how long it takes an individual to change sex, and then of course you have the behavior that can theoretically change much more rapidly than the gonads.
We have a lot
but-
to say about this. Yes, we do.
Yeah.
We could have a whole other
Totally.
seminar on this. I'll say a few
Mhm.
words on this just because I can't help myself, but we'll have to meet together to talk about it. First is that why I want to answer your question about the longevity. I think the biggest picture is that we don't really know a lot about protandry, and most what we know about sex change has to do with in protogynous species, and that's where fish start off female and become male. And in that system, people, I guess, just assume that it was sort of all the same, like originally, like in protogeny, they change behavior like immediately. Like as soon as they raise their status, they like start displaying copulatory behavior, like all the behavior, like nothing. You don't even need the gonads. They can dissect out the gonads. They still do the behavioral change. And then what happens is their gonads change very rapidly within a week or 2 weeks. And then the gonads, those are now androgens, and they release a testosterone, which then takes over and causes changes in their coloration, makes them larger, and then also is responsible for sculpting some of the POA changes that I described, particularly the number of cells, GnRH neurons have been looked at. Now, in anemone fish, it's totally different. So first of all, we thought they changed behavioral sex right away, but we just tested this empirically and proved that absolutely we were wrong. They do not change behavior right away. In fact, their behavior requires their complete sex change. And not only that, they have to now be ready to reproduce almost, it seems, because we have fish that were in the midst of sex change that actually they were like 6 months out. They had vitellogenic oocytes, which means they had yolked eggs. They hadn't laid eggs yet. They had their hormone levels were not totally female. They were on the way to go there. And we have a litmus test here because you can drop that fish in a tank with another female, and females basically are very aggressive toward other females, whereas males are not aggressive toward other females. And this fish was basically not very aggressive. It was aggressive toward females, but not nearly as aggressive as females are to other females. And it was also very aggressive towards males, and females are not aggressive toward males. But even better than that is the fact that you can give them eggs, and these fish will care for the eggs spontaneously, which is another whole story as to why they do that, but they do. And when you give sex-changing fish, even those fish that had vitellogenic oocytes, and you give them eggs, they're the one that go in and take care of them. That is a purely male feature, not their partner, who is the male, right? The sex-changing fish is much bigger, and so it's the one that should not be caring for it. It goes in and cares for the egg. So that is a male trait. So that means their behavior is totally different. Now, the gonads also are very different. In the protogeny, it looks like the gonads slowly march towards the process. So right away, they start getting androgens. That is not what seems to happen with anemone fish. Their gonads basically disintegrate, and it has only to do with the fact that they're not reproducing. Everyone knows that when fish don't reproduce, their gonads shrivel up, and that seems to be what's happening, because the same thing happens if I pair two males together, the subordinate male's gonads also look exactly the same as the female's gonads. And they can stay like that for years, okay? Years. And this is why I think it takes so long, because the brain has changed, but they're not yet going to tell the gonads to change. They're going to wait. What are they waiting for? I think they're waiting maybe to grow bigger. I think they're waiting maybe to feel like the night is right for the reproductive event. These are individual differences. I don't know what all those things are happening, but their brain is set up to do it, but their gonads are in this totally different stasis state, just waiting there. And that's what allows for the variability. And, these fish, they have to grow. They grow tremendously. When the females you could see from that picture, they're like, they could be very much bigger than the males. And so that takes a while to grow. If their male is much bigger, maybe it can happen faster. If the fish is smaller, it takes longer. So anyway, that's a long answer to the question, but it's very different between protogeny and protandry. And I want to make a big deal of that, because I think that's what's interfering with our thinking in this model. Behavior does not change first, it changes last. So, and gonads do not slowly march. They sort of have nothing to do with the process, really. It's almost like this whole thing is gonad independent. The gonad is just waiting there until the brain has all decided what it's going to do, and then it changes. So I'm probably way out. Oh, not too bad. So, but we should meet and talk more about this. And
Yeah, thank you.
I'll go over to your lab and see the sticklebacks. Or you can come, you should see the anemone fish.
Yeah, I would love to. We could talk later
Yes,
though,
see the anemone fish.
if other people have questions.
Yeah. Any other
Yeah,
questions?
thank you.
Sure. Everybody can come see the anemone fish. There's a place on 1805 South Wright Street, free parking. We've got about 2,000 gallons of marine aquariums. I've got a moray eel that's about 2 feet long, and I have all kinds of different wrasses, other fish that change sex other direction, and clownfishes that we've been breeding, and you know, transgenic fish and all kinds of messed up stuff. So come and see it.
That all sounds amazing. I'd love to check out at some point too now.
Yeah, check it
That's
out.
super exciting.
Yeah,
But yeah, if there's no other questions, thank you so much, Justin, for talking with us, and thank you all for joining us in for Science Cafe. It was great. I had a great time and I learned a lot.
all right. See you guys.